So we are now in the biology part of the test. 23. Reverse transcriptase is mainly found in retroviruses such as HIV. 4 nucleotides are needed for the reaction catalyzed by this enzyme. A group of molecules may inhibit this enzyme. These molecules are structurally similar to the nucleotides, but slightly altered so that bonds normally formed in the reaction cannot be made. Which of the following statements may be correct? The 4 inhibitors molecules will resemble adenine, thymine, guanine, and cytosine. If we think about the reverse transcriptase, what actually reverse transcriptase does, it takes RNA and it gives us a complementary DNA. If we have 2 complementary DNA, we can assemble a DNA molecule. And after the formation of the double-stranded DNA, we have the integrase, which is an enzyme that helps create the sticky ends, and the sticky ends will then be like integrase and integrase name told us, will integrate this DNA, this virus DNA in the host's DNA. So the first statement is actually true, because we, in order to transcribe RNA to cDNA, we need the 4 nucleotides, and those nucleotides will be adenine, thymine, guanine, and cytosine, because we are trying to make DNA. We know that in RNA we have uracil that is taking the place of thymine. But in this case, the 4 inhibitors will resemble actually adenine, thymine, guanine, and cytosine. For example, we have a strand of RNA that may be uracil, adenine, guanine, uracil, and cytosine. We know that uracil pairs with adenine, adenine with thymine, guanine with cytosine, in this case adenine and then guanine. So we formed our cDNA, complementary DNA, because it's complementary to the RNA. And at this point, if we, for example, switch this thymine with some strange inhibitor that has a structure similar to thymine, let's call it, I don't know, X. X can actually inhibit this molecule. Let's see if number 2 and 3 are actually two correct suppositions. The inhibitor molecules can prevent the formation of phosphodiester bonds. So what is phosphodiester bonds? It's actually this bond over here. It's between a phosphorus and the 2 oxygens. And this bond and this type of bonds actually 2 nucleotides. So let's say that nucleotide X has some strange feature that does not permit this type of bond. we can see that number 2 is actually a good statement. Number 3, the inhibitor molecules can result in a reduction in the number of the new virus particle produced. Yes, because if we prevent the formation of the phosphodiester bonds, we prevent the formation of cDNA, we prevent the integrate this DNA into the host's DNA, we can see that the host DNA will not produce virus particles. And so if we reduce this all of this process, we can actually reduce the number of particles. So number 3 is also correct. So the answer, or our answer in this case, is C, that every statement is correct. The enzyme HIV protease is produced by the HIV virus. Okay, so this 2018 IMAT is actually around the HIV virus. So the enzyme HIV1 protease. So we know that protease inside the HIV virus helps cleave some proteins, helps essentially create some functional proteins, because it cleaves off some residues that may not be important for the virus. The enzyme is formed from two identical chains of 99 amino acids. In each chain, amino acid 25, 26, and 27 in the sequence form parts of the active site. So, which level of the structure control the shape of the active virus of the HIV protease? So we have four levels of protein structure. We have the primary, the secondary, the tertiary, and the quaternary. So the primary is the sequence of amino acids. The secondary is how these chains of amino acid actually communicate between them. We have two types of communication. We have the first type, that is a beta sheet, and then we have an alpha helix. So we have bonds between, for example, nitrogen and nitrogen, oxygen and nitrogen and hydrogen. So they are kept together by hydrogen bonds. The tertiary structure is between those units and is a 3D structure, and the quaternary structure is between subunits. So subunits 1, 2, 3, and 4. For example, hemoglobin has 4 subunits and an heme group in between them. Okay, so if we we have HIV protease, and which level of structure control the shape of the active site? Okay, so the answer is letter C, because every level of structure actually controls something. If we change the primary structure would change everything else. If we change the quaternary structure, the protein would not be functional anymore, even if we change the tertiary or the secondary. So in this case, it's letter C, because it's everything.